is Ran Lee, and he will be speaking on advancing AAV manufacturing through scalable cell line development, high cell density transfection, and robust purification.
Okay. Good morning, everyone. So thank you, Frank, for the introduction, and thank you for the opportunity to present here of our work. So my name is Ran Li. I'm leading upstream process development and analytical development here at PackGene Biotech. So today we have 4 topics. The first one is business introduction. Second one is we'll discuss our effort on upstream process intensification with high VCD transfection. Third one is just showcase our downstream optimization with a 2-step AEX purification to increase full vector, for enrich the full vector population, and then follow with a question and answer. So first, our business introduction. So we are a CDMO. We provide a one-stop CDMO service to our clients from AAV development to manufacturing. We have both DNA, plasmid DNA, and also viral vector production platform. For DNA, we can produce plasmid. We have a 200-liter scale and can deliver 100 gram per batch. For viral vector, we can scale up to 1,000 liter, and we have both adherent and suspension systems. And we also can do most of the analytical testing in-house. We developed over 60 QC release assays for our customers so far, including some challenging ones like TCID50, RCAAV, and a full empty ratio characterization with either AUC or mass spectrometry. So of course, all the operations are overseen by QA to ensure GMP compliance. And we have a facility in both Guangzhou, China, and also Houston, Texas. So in Guangzhou, we can produce both GMP vector and plasmid. And in Houston, with a newly built 35,000 square feet facility, we can do we focus on the GMP vector production. So far, we support over 150 we already delivered over 150 GMP batches and support our customer over 32 IIT trial in China with over 11 IND filings successfully in the US. So there is a seamless tech transfer between China and US facility. So a lot of our customers begin business with us from as early as discovery stage. We provide CRO service for target identification. Then the China facility will provide a GMP-grade virus to support their trial, IIT trial in China, and can get a very early clinical data. And if successful, and the client will tech transfer the process from China facility to Houston, then we can produce GMP-grade virus to support a clinical trial in the US. And then if upon approval, we can also further support their commercialization support needs. So next, let's go to some science topic. The first one, the topic is upstream process intensification with high VCD transfection. So most 3-plasmid transfection happened in industry at a very relatively lower VCD, about 2 to 3. Let's see, later. Anyway, so there's around 2 to 3 million per mil. And we are trying to achieve a higher VCD transfection of push to about 10 million per mil or even higher to 20 million per mil. Ideally, you want to triple or if you want to triple your transfection density, and also you want to triple your yield. With that, besides the yield, the benefit of a high VCD transfection, you can also lower the cost per dose. For a single dose, if you achieve high VCD transfection, you can also reduce your footprint. However, there is a lot of challenge for this high VCD transfection. The most significant one is well documented in literature that while the transfection high VCD, your cell began to lose productivity, and also you have a lower transfection efficiency. And that phenomenon is well known as a high VCD cell density effect. And of course, to push the cell to a higher VCD to transfect, you also need a perfusion process to support that. And also, there's more cell stress at a high VCD growth and transfection. And also, the process requires more optimization or more careful control. So to address this, to overcome this high VCD transfection, this high density effect, So the first, we address this lower efficiency by optimizing our lower transfection efficiency by optimizing our transfection complex. So the work, we significantly improved our transfection efficiency with just a complex DNA transfection ratio and also volume. So I won't be able to show the work here, but the more challenging one is to still, even with that, the cell still cannot get the same productivity. To address that, we determined to add different cell feed or cell boost to increase the cell productivity. So this slide showing our process design. So first, we screen the different cell boost to see which one can really increase the cell productivity. So our upper left showing our just control. We transfect the cell in this scaled down model, the shake tube, with a 3 to 4, about 3.5 million per mil. And after 48 hours, we harvest. That's our low VCD transfection control. We should get the same productivity as that tube. And then for the high VCD, we grow the cells in shake flask, then concentrate them to 10 million per mil. Ideally, we want to see triple the yield. And then there is a theory that this loss of cell productivity is caused by this build-up waste material at high VCD. So we performed the top arm is showing we do a medium change after 24 hours, try to do a mimic perfusion, and then to get rid of those toxic metabolites, and harvest another 24 hours later. The bottom one, we add different cell feeds at 24 hours without perfusion. So we screened about 12 commercially available 12 feeds. You can see 5 of the cell feeds, 1, 2, 5, 7, 12, that really can increase the cell titer. And the low VCD titer is shown in the blue bar, and the perfusion process showing this medium change, which mimic perfusion, shown in the red bar. You can see that really the perfusion, the process could not really rescue this loss of cell productivity. And since there is a dilution effect when you add feed, so we further calculate the yield, try to get more accurate data. And you can see again the top five feed is 1, 2, 5, 7, and 12. So we'll further verify this result in a shake flask study. So, and the design is almost the same, but we just switched the tube to a higher volume, about 30 ml shake flask. And you can see it again, the titer, we get a very high titer, about 3E12 Vg per ml, with the feed from number 2 and from number 2 and number 7, sorry, number 2 and number 12. And low VCD, you can see on the middle bar, and in the middle graph, low VCD about 1E12, and with a triple the cell density, we got a triple the yield. So consistent with that, we can see that we also normalize the yield to the transfection cell density to get the cell productivity. We can say that feed number 2 and number 12 really can rescue this loss of cell productivity at high VCD. So then we next quantify, analyze the quality of the vector from those conditions. To get the full empty ratio, so we developed this simple assay, which can allow us to analyze any product with no matter how dirty it is. We can just, and also you require a very small quantity. So this one, we just use a small 100 microliter your in-process sample. Again, can be clean, can be dirty, but we put down the AAV from using the magnetic dynabeads, and then after following wash and elution, we can just we can analyze full empty ratio with mass photometry just with 10 microliter of your samples. And you can see that we can get a full empty ratio from our upstream cell lysate. Again, the green bar showing the full empty, full vector ratios. Most of the cell feed can deliver acceptable level, 30% or more, a full vector at the beginning. And the number 5 actually has the highest full vector ratio, but unfortunately that has a lower cell productivity. So the number 12 and number 2 and number 12 is our top candidates. But considering number 2 is a customly made cell feed, so we decide to move forward with the number 12, which deliver about 30% of full vector at the beginning. And to scale this one to a higher process, now we cannot do medium, just spin down, centrifuge and resuspension to 10 million anymore, so we have to use a perfusion process. to push the cell to a higher VCD. So this is just, we just do this simple testing, leverage the perfusion function of the wave bioreactor. First, we grow the cells to about 2 million per mil, 2 to 3 million per mil, then we start the perfusion process, about 1 vessel volume per day. And then we push the cell VCD to about close to 10 million. In this case, we transfect around 8 million per mil. And then after transfection, we add the feed number 12 on transfection day 1 and day 2, when we harvest on day 3. And each day we take sample and check their the variability, just productivity. You can see the most, the cells, again, it's consistent with the shake flask data. We can see that both day 2 and day 3 can really get the same cell productivity as the shake flask or the low VCD transfection conditions, indicating this process is fully scalable. We get a high titer about close to 2.8012. So again, if that's successful, in future, that's our goal is hopefully we can push the high VCD to an even higher range, to 10 million or even 20 million. Then 1,000 liter transfection low VCD, we can simply just do a 200 liter transfection high VCD. That's significantly improve, just to reduce our footprint, cost, and time. So before going to the last topic, I just want to jump quickly to this, just introduce our two plasmid system. We have our engineered wrap and cap plasmid in one plasmid, and then with GOI, transfect the cells, and then we have specially designed enhancer, AR22, 30, and 35. And then with that, that one can significantly reduce whole cell DNA and also increase the yield. You can see the two plasmid system generally have better yield quality and robustness, and also based on data have with one plasmid less is a lower cost. You can see the lysate titer. This is AAV9 GFP. 3 years ago, when I presented another conference in Boston, our titer record is 3E12 VG per mil. Now this one, you can see we already get a 6E12 VG per mil. That's super high, I think, across the whole industry. So, and then that's also not just limited to one single transfection reagent. You can see the two plasmids, we can do a 3 GMP-grade transfection reagent, and 2 of them are very commonly used in research and industry. So we don't want to tie our customer to just single one transfection reagent. And also residue whole cell DNA, we can, with the enhancer number 22, we can push the residue whole cell DNA to single-digit nanogram per E13 VG. That's also true for all the transfection reagent. So due to, I think, the time runs out quickly, just we'll have 2 more slides. Just introduce quickly about the downstream optimization with two-step AEX. Sometimes your starting material may not have a good quality. You have a relatively lower full vector ratio. You purify with through AEX when you reach full with a collection criteria with 1.2. And sometimes you still could not get a very good quality of full. In our case, sometimes we got less than 60% full. However, instead of do an extensive optimization, we decided just to AEX, first collect the sample at 1.1 to enrich more full vector, but also just get rid of some empty vector with a lower quality collection criteria. And then the next AEX, we can do a higher quality with 1.3 ratio, 262 versus 280. Now with that, we can enrich the vector to over 80%. And with a client project, it's a real client project from 3 liter, 15 liter to 100 liter scale up. We got about the titer, upstream titer across about the same. But you can see the downstream purification. If with one-step AEX, we can get about 60% full vector. But with this 80, we got this, with two-step, we can get almost 80%, like over 80%. And for the 200 liter, we got over 90% full vector with this two-step AEX enrichment. Again, two-step does not increase the time significantly, since a monolith column can get a very fast flow, about 2 hours each round. So adding another round doesn't really increase the, we can still get finished AEX step in 1 day. So I think that concludes my talk, and make gene therapy affordable is our goal. And with that thought in mind, that drives our effort on those process intensification and process optimization. So with that, I really thank you for your time and ready for any questions.
Yep. All right, go ahead.
I have a question about your perfusion samples.
Okay.
So did you increase the plasmid amounts and transfection mix proportionally? You know, when you increase the cell density to 8 million, you know?
Yeah, that's actually, we could not disclose that, unfortunately. Yeah.
How about empty to full ratio for the intensified, you know, for the perfusion the sample?
Yes, actually, it's shown here. It's actually the I would say the perfusion, actually not shown here. The perfusion process have about similar full empty ratio compared to low VCD. Yeah.
Thank you.
So it's but the titer could not reach like a threefold. You increase the VCD threefold, the titer could not. But the full empty ratio are comparable between high and low VCDs.
Thank you.
Yeah.
Okay, next one. Go ahead.
Hi, Ryan Maduresh from Sanofi. Nice talk.
Thank you.
I was wondering about the data that you showed with the feeds intensifying to 10 million. Those are not with perfusion, right? Just for clarity.
Those are not, no, because we try to reduce the cost of perfusion. So.
And how about if you use perfusion with those feed, do you get additional benefit? Because generally we see in cell density effect that the application of perfusion increases the cell-specific productivity. What's your thoughts on that?
Yeah, so that's actually, thank you, that's a great point. So on I think the top arm, we try to mimic the perfusion just by medium change. We could not really get like the red bar, actually is, in our case, we could not really get the fully recover of this cell productivity. So that's the reason we go to another approach, just add feed. We just perfuse, go to a higher VCD, and then we stop perfusion, just add feed, follow just afterwards. And now we can see we can successfully get a 312 VG per mil with this approach.
So essentially, post-transfection, you're able to maintain cell-specific productivity by just using a fed batch process.
That's correct.
Thank you so much.
Thank you.
Please help me thank our speaker.
Thank you for your time.